Hello, everybody, and happy St. Patrick's Day. I know it doesn't feel like it. We're not in class together. This is actually my basement. This lecture is specifically for 6th and 7th period who didn't get to see how to go through the spreadsheet and have a practice run with one of the groups of living things that you're going to do your project on. So I'm going to do this lecture specifically for 6th and 7th period, but I will share it also with 3rd and 4th so that you can have it to remind you of what to fill in and maybe help you with your projects as you move forward. But of course, don't be afraid to ask me any questions via email. So here's your fact of the day so we can get started, and that is that koala bears actually have fingerprints almost identical to humans, and that there have been times where koala prints have been found at scenes of crimes and have thrown off investigations. So there's your fact of the day. Let's get started. The top row of your spreadsheet, I've also shared this via Google Docs if you want to do it digitally. The top row is bacteria. This one is not a typical group. Of course, it's not an animal, it's its own kingdom. So that's why I'll tackle this one, because there's a lot of ins and outs to it. But I will help you with what to write on your spreadsheet and show you some stuff about bacteria, because forget all this virus talk. Viruses are lame and boring. Who even knows if they're living? Bacteria, they're the real deal, and there's actually a lot of good bacteria, even though most of bacteria gets a bad rap. So let's get started. Okay, first piece of information that you can fill in that's also right here is that bacteria are single-celled, right? So the first square of my row, bacteria, is are they single or multi-celled? Bacteria are all single-celled. They may be found together, but those are just colonies of independent cells not working together. Also on your sheet, some other basic things. Since they are single-celled, they don't work together and have any fancy structures like a spinal cord or an internal or external skeleton. So you can just write NA, not applicable, or none. That would suffice, right? As you can see, the squares are very small, so I'll help you with just writing 1 or 2 word answers so that you're not trying to cram in a whole sentence. Moving on. This slide is to show you what I don't want to see on your projects. This is my least favorite thing, is when you put text over a picture. And so I used a really pixelated picture and some really dark text just to overprove my point. So when you're working on your projects, please don't do this. Okay? But it does show, which is hard to see, is how we classify bacteria. One of your slides will talk about classification, not just filling in those squares. And so for my part of classification, bacteria is broken up into 2 groups, the eubacteria and the archaebacteria. As we said, they are actually from 2 different domains, archaea and bacteria. Other ways that we classify bacteria is by their shape, whether they're spirals or rods or circles. We also can classify them by whether they're gram-negative or gram-positive. Some of you who have parents who are in the medical field, you've heard of gram staining and gram stain tests. Sometimes bacteria is classified by how they move, whether they're mobile or motile at all, whether they have flagella or pili or things like that that help them move. They can also be classified by how they obtain their food or energy. There are heterotrophic bacteria, which are consumers, either parasitic or they are just predatory. There are autotrophic bacteria. They either make their own food through photosynthesis or they manipulate chemical reactions. Those are called chemoautotrophs. It's hard to kind of read there, but chemoautotrophs. This will all be shared with you, of course, to check out. They can also be classified by how they carry out their respiration. Most of them, of course, are all anaerobic, and so some of them are obligatory anaerobes. Those were the majority of the ones that died off during that first mass extinction because oxygen is a poison to them. But the rest of these guys can tolerate it, oxygen that is. And so that's how they can be classified as well. They can be classified by what they consume. Some bacteria consume methane, that stinky gas, right, that comes out of the business end of a cow, or halophilic, right, short term for salt loving, and they can feed off of that as well. The bacteria that is autotrophic, that carries out photosynthetic, has a fancy name, cyanobacteria. Okay, back to your slideshow. I just wanted to include some extra information. Bacteria is a very broad subject. I could spend the rest of the year talking about it, but let's get to the matters at hand. So, when you get to your slideshow, when you're talking about nervous system, circulatory system, things like that, right? The single-celled bacteria doesn't have fancy structures like yours will. So for the nervous system, you can just write DNA controls the cell. For a circulatory system, materials just naturally circulate, so you can just write the word natural for circulation. Digestive system, it doesn't have a digestive system, but it does have, I hope you all said enzymes right there, right? Enzymes, so you can just write the word enzymes for your digestive system. Other body parts, they don't have lungs, but materials enter and exit through a process called, I'm hoping you just said diffusion, because that's the answer, so write the word diffusion for respiratory system. Sense organs, same deal, no eyes, ears, nose, tongue, things like that, but their cell membrane can detect certain chemicals, and so for the sense organs, you can just write cell membrane. Okay, reproduction is a tough one. They do the normal mitosis. They can conjugate, which means share some DNA, but that is rare. So I'll show you what to write for that. The last square on the end asks for an example. Here's one of the bad ones, right, where it's a pathogen like leprosy, but strep throat, pneumonia, staph, E. coli, syphilis, and tuberculosis are all bacterial infections. Many bacterial species are good. They help us in the process of genetic engineering. They help us in making food like yogurt and other dairy products related to that. My favorite, of course, are the nitrogen-fixing bacteria that fix nitrogen back into the soil. That's why we plant soybeans around here every so many years, because they help to re-nutrify the soil. So bacteria, not all terrible. We use it, like I said, for its ability to help. Don't worry, I'm just coughing because of the allergies. I don't have any coronavirus. Bacteria is also used for genetic engineering. We have bacteria in and on our bodies, which I've stressed many times, that help us to make vitamins, digest food, kill pathogens, you name it. Bacteria is great. So, one more slide to help you round out the last few squares on your slide. This talks about warm-blooded and cold-blooded. You could put NA. Most of the time they would be considered cold-blooded because they move slower in the slower temperatures. They do not lay eggs. Their fertilization is not internal or external. Um, they do not nourish with milk or care for their young. As you guys go through your projects and you see more and more species get complex, you'll be saying yes to a lot of these things, but bacteria, very simple, doesn't do this stuff. So of course, I can't end without giving my salute to bacteria. Feel free to replay this. If you didn't get all the squares filled in, also feel free to email me with any questions concerning this lecture or any of your projects. Thank you, and be well.